right. Hi, everybody. Thanks for tuning in today. This week, our seminar speaker is Dr. Rika Anderson, who is currently an associate professor at Carleton College, which is in Minnesota. Dr. Anderson is an astrobiologist whose research focuses on better understanding how microbial populations adapt and evolve in habitats that are relevant to the study of astrobiology, for example, deep sea hydrothermal vents, which I know she has spent a lot of time thinking about. She received a bachelor's degree in biology from Carleton College, and then a master's in oceanography and PhD in oceanography and astrobiology from the University of Washington. After that, she was a NASA astrobiology postdoc for several years at the University of Illinois at Urbana-Champaign. Later, moving on to a postdoc at the Marine Biological Laboratory at Woods Hole. And in 2017, she started her position there at Carleton College. And so thanks again, Dr. Anderson, for taking the time to talk with us, and take it away.
Cool. Yeah, thank you for the invitation. It's really fun to get to talk to you guys. I know a lot of, I know the Georgia Tech astrobiology group is really awesome. So it was great to be invited. So let's do the usual Zoom thingy where I share my screen and then push play and hope that you all can see this. I think you can. If not, somebody just yell, but I think that should work. Perfect. All right, so I'm going to start off just by giving you a broad overview of what it is that my research group does. And I should give you all a big caveat. I'm at a small liberal arts college at Carleton. So all of the students in my research group are undergrads. And I'm actually really proud of that. They are awesome. Some of them may have gone to Georgia Tech afterwards or may soon go to Georgia Tech. I'll certainly be pushing that. But it's just something I wanted to highlight because I'm actually really proud of these students. And so what we do in my group, in terms of the big picture questions we think about, as Frances said, I very much think of myself as an astrobiologist in a big picture way. So we think about these big picture questions like, how did life first originate on Earth? And then once it originated on the planet, how did it then start to spread across the planet? How did it diversify and start to colonize new ecological niches? And more fundamentally, how do life and its environment co-evolve over time? Now, I realize those are very broad, big picture questions, and I'll start to go into a little more detail in a moment. Usually when I give talks like this, I'm able to use videos like this as a crutch because they're pretty. And if you guys start to get bored, I just throw a pretty picture up there or a video and it attracts everyone's attention again, and you can ignore the stuff coming out of my mouth. This is because a lot of the work that I do is focused on thinking about microbes in deep sea hydrothermal vents. As an astrobiologist, I'm interested in them because some have proposed that hydrothermal vents could have been a site for the origin and early evolution of life on Earth. And so one of the things my research group does is use submarines to go down to these vents, collect samples of the microbes there, and try to understand how they evolve over time. But here's a big but. Appreciate this video while you can, because I'm actually not going to be talking about my hydrothermal vent research today, because I know you all are an astrobiology group, and I actually have some recent projects that are much more astrobiological. What are the size of these vents? These are at a place called Picard off the Mid-Cayman Rise. I would say that the diameter of this vent is maybe this big, so they're not that big actually, and they're maybe 2 or 3 meters tall. And these actually got knocked down by an earthquake the day after we saw them. So they're actually pretty fragile as well. But like I said, appreciate the vents while you can, because I'm actually going to be talking about some other projects that I think are actually almost more astrobiologically relevant, related to the early evolution of life on Earth and the coevolution of life and its environment over time. So say goodbye to the hydrothermal vents. I'm happy to talk about them if you all have questions, but I'm actually going to move on. And we're going to be pulling back a lot and thinking about Earth in a much broader context. So for those of you who don't recognize this image, this is one of the pale blue dot images that has come from satellites that have gone far beyond the Earth to circle back and look at the Earth from, a very big scale context. This is not the actual original pale blue dot image that Carl Sagan asked NASA to take using Voyager. This is a more updated image where you can see the pale blue dot that is Earth a little more closely. But I like to show this because, this is Earth in its solar system context, in its universal context. And as astrobiologists, we're trying to understand that pale blue dot and how it evolved over time, how life changed that pale blue dot. Was it always blue? And can we use that information to then find other pale blue dots elsewhere? That's really what we're trying to do. And as one of the ways to do that, we're trying to understand potential biosignatures as we look at exoplanets around distant solar systems. So a biosignature, broadly speaking, is any substance or phenomenon that provides scientific evidence of past or present life. And it has to be a gas that's produced by life that is detectable using remote telescopes. So some example biosignatures are things like oxygen. We all know that oxygen, for the most part on this that we're breathing, is produced by life. That's a key one, but there's also things like vegetation can create a characteristic red edge in a spectrum. That is a sign that you have a forest-covered planet, for example. Water is detectable, but obviously not always clearly a biosignature. Methane is another one, that carbon dioxide and methane together form a pretty distinct biosignature that's actually detectable using the Webb telescope. So that's one we'll be looking at in close detail in the coming years. And there's many more besides this. But what we're trying to understand is, what are the gases produced by life? How are they created? How are they destroyed? When were they created at different points in Earth's history? And can we detect them using these telescopes? And of course, a planet that's alive, that has life on it, is going to produce a different set of gases than a dead planet. So the graph on the right, you can see an example of different types of gases produced on Earth and the surface flux of those gases compared to an abiotic planet, just in a modeling scenario. And the figure on the left shows predicted gases produced on Earth, Mars, and Venus. So you can see that we'd expect different sets of gases and different fluxes of those gases on different planets, because some of them have life and some of them don't. And then, of course, if we look at Earth itself over time, the biosignatures on Earth would have changed as well as life evolved on Earth. I love this figure. This was a figure generated by some colleagues of mine, Jada Arney and Sean Domagal-Goldman at NASA. And what they're showing here on the left-hand side is different fluxes of gas over time: oxygen, water, ozone, methane, carbon dioxide, the ones I highlighted in my last slide. And then they're showing, if you were to look at a spectrum of the Earth, you can see how it would have shifted from the Archaean to the Proterozoic to the modern Earth. And so if we want to understand, when we're looking at a distant exoplanet, what is this mixture of gases that we're detecting? What is the spectrum telling us? We can use the Earth as sort of a case study for how we might expect different mixtures of gases to evolve over time, to understand these biosignatures, what gases tend to emerge when and in combination with each other. So I spend a lot of time working with astronomers to say, okay, what can you detect? What are the gases that you think are the best ones to target? And then they ask me, okay, well, how would these gases have changed over time? When would they have emerged, and so on? So I spend a lot of time talking to astronomers and asking really dumb questions, and we kind of go back and forth, and it's a lot of fun, actually. No talk about the early Earth would be complete without showing this figure, so check. What I'm showing here is the amount of oxygen on Earth over time. So we have 4 billion years ago on the left axis, on the left side of the x-axis, present day on the right, and you can see that we would have had very little oxygen on the Earth, of course, until the Great Oxidation Event, which you can see in that jump in oxygen that you see about 2.3 billion years ago. And then, of course, life, through the form of oxygenic photosynthesis, would have produced that oxygen, which would have then, increased the amount of oxygen in the Earth's atmosphere. To put a little more context to this image, you can see here we've reproduced, by we, I mean somebody else has reproduced that curve of oxygen on this graph and pinpointed some key events in the evolution of life on Earth over time. So we think the origin of life would have been about, depending who you ask, 3.5 to 4 billion years ago, somewhere in there. Then the first oxygenic photosynthetic organisms would have emerged, again, depending who you ask, some point thereafter, 3.5, 3 billion years ago. The Great Oxidation Event, about 2.3. And then we would have had the first eukaryotes appearing, ignore the boring billion thing, thoughts about that, and the first animals appearing later on. All right, so we are trying to understand, again, as Earth changed over time, as the planet evolved, as the amount of gas produced by that life changed over time, we want to get a closer look at that to help give the astronomers information they need to then model the kinds of biosignatures you might expect to see around other exoplanets. So some of the big picture questions that I've been asking lately are, which gases are microbes producing at different periods of Earth history, and when were those gases released at different periods? So we can kind of get a sense of, when did different metabolisms appear? When did different metabolisms become ecologically important? And were they found in combination with other metabolisms that might give us some sort of distinctive biosignature signatures that we could see in spectra if we were to look at the Earth from a distant telescope. And then, of course, like I said, in working with astronomers, they always say, okay, Rika, that's great. I'm glad these microbes are producing these like weird gases, but are they actually detectable? That's one of the big questions they have, because if you have a microbe that's only producing like 2 molecules of some gas, and there's only a tiny colony of those microbes living on one part of the Earth, the astronomers are like, that's great, I'm happy for you, but I'm not going to be able to see that with a telescope. They keep me grounded. So I'm actually going to focus on nitrogen, the nitrogen cycle. This is a project that we did in the past couple of years in my lab, and we care about nitrogen for many reasons, but one of the most important ones is that it is a limiting nutrient on Earth. So it has a major control on biological productivity on Earth. Over time, you can see this is a simulation of life in the oceans and on land, and you can see how it changes over the course of the seasons. Nitrogen is going to be one of the, not the only one, but one of the limiting nutrients that affects how much biomass, how much primary production occurs in these different habitats over the course of a year. So nitrogen is important for that. Some have suggested that nitrogen could have been a key nutrient in the rise of eukaryotic algae. So if you care about eukaryotes and the rise of complex life, nitrogen could be an important piece in that puzzle. The abundance of nitrogen, especially N2 gas, is thought to be linked to the amount of air pressure on the early Earth. This is one of my favorite studies, actually, by a colleague of mine. They were trying to figure out how much gas was in the atmosphere, how much atmospheric pressure was there in the Archaean, and they determined that by measuring raindrops, fossilized raindrops in Archaean sediments, which is crazy to me. And that became a proxy for basically how much nitrogen there was in the atmosphere in the Archaean. N2O is also a greenhouse gas, and so the amount of N2O in the atmosphere would be linked to climate as well. And I also want to mention briefly that N2O is thought to be a potentially important biosignature, albeit potentially a weak one. I hear this from my astronomer friends. This is a figure from a colleague of mine who shows the spectral signatures of different biosignatures, just so you can get a sense of what they would look like. And N2O I've highlighted here as an example of one of the biosignatures that they claim is potentially detectable using telescopes, depending on how much is actually being fluxed from life on a given planet. So the nitrogen cycle is important for that reason as well. It produces potentially detectable biosignatures. So here is one view of the nitrogen cycle produced by my colleague, Eva Stucken. Most of the different steps in the nitrogen cycle are driven by microbial metabolism. So microbes seeking energy are responsible for most of the steps in the nitrogen cycle, driving these steps forward. Different steps in the nitrogen cycle occur in higher oxygen and lower oxygen environments, and those are highlighted in different colors here, where the black transformations tend to occur in more anoxic environments, blue are oxic environments, and orange are more suboxic, so lower oxygen environments. And so you could imagine that because the modern nitrogen cycle, we have different steps occurring in different oxygenated environments, you might imagine that as the Earth's oxygenation state changed over history, we might expect to see the nitrogen cycle to evolve over time. And in fact, here is a visualization of that by, again, my colleague Eva Stucken, imagining how we might have seen the nitrogen cycle change over time from the top left when we had very little oxygen present in the atmosphere and in our oceans. And then as oxygen started to appear on the Earth, we might expect to see the oxygen cycle to evolve over time and become more complex as the steps that occur in oxic environments began to emerge and become more common on the Earth over time. Most of what we know, but a lot of what we know about the evolution of the nitrogen cycle over time comes from geochemical data, from isotope data. I'm going to give a big caveat. I'm a biologist and I am showing isotope data, and this is scary, but this is how we know what context we're operating in. So this is an image showing nitrogen isotopes and 15 isotopes over time. Again, we have the early Earth 4 billion years ago on the left side of the axis, modern day on the right side. They've superimposed our favorite oxygen line on top of it. And basically what they wanted you to show is that the isotope fractionation seems to have shifted after the great oxidation event. So you can see that there is less negative fractionation going on after you hit the great oxidation event, suggesting that we would have seen an onset of aerobic nitrogen cycling going on after the great oxidation event, perhaps unsurprisingly, but there's data in the geochemical record to support that. One big question that many people have had about the nitrogen cycle is, when did biological nitrogen fixation first arise? Nitrogen fixation is the process of taking N2 gas out of the atmosphere and turning it into a biologically useful form. That is really hard because N2 gas has 3 bonds connecting the 2 nitrogens. So it takes a huge amount of energy to break that triple bond and turn it into ammonium, which then makes it available to the rest of biology in the nitrogen cycle. Some have argued that that ability to fix nitrogen came super early. Some have argued that it came much later. My colleague, again, Eva Stucken, I mention her a lot because she's actually my partner in crime and has done a lot of this work with me. She's a brilliant geochemist. She did some isotopic work looking at, I think, shales from about 3.1 to 2.7 billion years ago. And what she found was fractionation up in the little box inset in the top right corner, showing that we had slightly negative delta N15 values occurring at around 3 billion years ago. And what she argued was that it's hard to explain that using abiotic nitrogen fixation, that this most likely was the result of biological nitrogen fixation very early in the Archaean. This was a paper that came out in Nature, and it was really the first geochemical evidence we had that that process of biological nitrogen fixation, which is so important in the nitrogen cycle to pull nitrogen out of the atmosphere and make it available to life. It was the first evidence really that that had happened so early from the geochemical record. So I've been showing you geochemical data, and that is a really important piece that we use to understand the evolution of life on the early Earth. The slight drawback is that you cannot reliably distinguish between all the different metabolic pathways. You're just looking at the rock record and trying to interpret it based on sort of bulk nitrogen isotope values. And sometimes you can't distinguish between the different metabolisms that are using that nitrogen. And it really only preserves evidence of the most dominant metabolisms. If you have a metabolism that's occurring in sort of lower levels, it may not get preserved in the rock record. If we combine that with using the genomic record, so peering into the genomes of microbes and using that as a record to see what was going on billions of years ago, we can actually distinguish between different metabolic pathways because we're looking at a gene by gene basis. Although we can't necessarily tell you directly how much of a given metabolism was occurring. That's where the rock record can help out. So I guess what I'm trying to tell you is that by bringing together the rock record and the genomic record, we might be able to gain new insights into how these microbial metabolisms may have evolved over billions of years and how they may have changed the planet over the past few billion years. So that's why I team up with Eva. She's a geochemist, I'm a biologist. I ask her all kinds of dumb questions all the time, and she very kindly answers my dumb questions. And we sort of work together to think about how can we address some of these big picture questions. Find yourself a rock friend, is my advice. So this is my view of the nitrogen cycle. I'm showing you here the different pathways. So nitrogen fixation from N2 gas down to ammonia. I don't think I have a cursor, so I'm just going to gesture, you can see it. And then we have nitrification from ammonia up to nitrate, and then denitrification from nitrate back all the way down to N2 gas with several other transformations in between. Again, most of these pathways are mediated by microbes seeking energy. And so we have specific enzymes, key enzymes that we can use as a marker for different pathways within the biological nitrogen cycle. So that's what I was looking at. Rather than isotopes in the rock record, we were focusing on different enzymes within the nitrogen cycle. So one important thing to know in order to understand how we did this is that genomes are not static. They change over time. So genes can be born through a variety of different processes. Genes can be duplicated, so you can make copies of genes. And genes can certainly be lost over time as well. You can have large-scale gene deletions, and you can have genes just disappear from genomes. That actually occurs fairly frequently. And you can also have a process called horizontal gene transfer, where genes can get horizontally transmitted from one organism into another that's not necessarily related by vertical descent. This can occur through a few different mechanisms. You can have viruses doing it that are shuttling genes from one microbe to the next. You can have microbes just taking up DNA that's lying around in the environment. And you can have conjugation, so where you have microbial sex, basically. They form a pilus and they can exchange often plasmids between different microbes. And the other thing to know is that modern metagenomics work, modern genomics work on microbes often find that there is pretty strong selection to keep genomes fairly streamlined, depending on the environment, depending on the microbe. So often a gene isn't necessarily retained unless it's useful, otherwise it gets kicked out of the genome, at least based on what we've seen in pangenomics work today in modern microbes. So we use that information to try to understand the evolution of these genes over time. A lot of previous phylogenetic work asks the question, when were specific genes born, which is of course very useful. When did the specific gene arise? Because once it arises, then microbes have the ability to, catalyze a certain metabolic pathway. But we also wanted to understand not just when genes were born, but when did they become ecologically important? And so let me give you a kind of dumb example. You've probably seen this meme. One thing to note is that this meme, which has become very common over the past couple of years for perhaps obvious reasons, unfortunately, this meme was actually created, I think it was in like 2012 or something, or maybe even earlier than that. But it didn't actually become ubiquitous and shared like crazy over social media until the past couple of years, again, for perhaps obvious and unfortunate reasons, because environmental conditions changed. And so in a way, when this meme was born and when it started to be shared widely on social media were two different things. And when it started to be shared widely, when it went viral, was a reflection of environmental conditions, i.e. a pandemic, and God knows what else has been happening over the past couple of years. And so we're using something similar. What I'm showing you here is a very goofily drawn graph where we have time on the x-axis, again, 4 billion years ago on the left side, modern day on the right side. On the y-axis, we have the number of horizontal gene transfer events, so basically genes getting shared between microbes. A given gene can be born at a specific period in time, but it's possible, so like it could arise on the tree of life at a specific point in time. But it's possible that that gene may not have been widely shared or may not have diversified or may not have been, horizontally transferred among organisms until it became more ecologically important, until environmental conditions changed to make that gene ecologically useful. So we were interested in tracking both these things, both when a gene was first born, but then also when it started to diversify and become horizontally transferred across genomes on the tree of life, because that might give us clues as to what might have been going on in the evolution of these genes over time. A similar use of this method has been done on a broad scale, and what they found was an Archaean expansion for all metabolic genes. So they oriented their graph a little bit differently from me. They have time on the y-axis, 4 billion years ago on the top, modern day on the bottom. They have gene loss on the left side and gene gain, so horizontal gene transfer and gene birth and gene duplication on the right. And basically what I want you to see is like this weird popping out that goes on around 3 billion years ago, where we have tons and tons of genes being born, being horizontally transferred, and also then being lost. So it seems like based on this study, they found there was this huge period of genomic innovation that happened around 3 billion years ago in the Archaean. And they were using this method to try to track when genes were born, when they were lost, when they were transferred, and found this really cool trend. So we wanted to do something similar, but rather than taking the very broad brush approach that they did in this study, we wanted to focus on the nitrogen cycle specifically to see if we could gain insights into the evolution of those genes over time. How do you do that? Okay, well, very broadly speaking, you have to start off by making a tree of life, which is no simple matter. We made a tree that has about 800 genomes in it, which takes about 2 or 3 months to run on our servers. And you want to make a tree of life that's anchored in time. So we call that a molecular clock. So we took fossil calibration points, so fossils from the rock record, to say we know that this kind of microbe had to evolve by at least this point in Earth history. And we use that to kind of anchor the tree in time to figure out, roughly when these different lineages may have arisen. I'm going to give you a giant grain of salt and say, just because of the nature of the data we have, the nature of the fossil record, the fact that life is incredibly diverse, these methods are necessarily imperfect. And so we have to proceed with caution in how we interpret these kinds of trees and these kinds of molecular clocks. There are big error bars because they're based on imperfect models, they're based on the limited data that we have. So one thing I'll emphasize is that rather than focusing on saying, okay, we know that this lineage based on the molecular clock arose at this period in time, I try to stay away from that. We want to think more about relative timing. Okay, it looks like this lineage or this metabolism may have arisen before this one. That's the kind of way that I think is a bit safer in interpreting these kinds of data sets. Okay, so we made a giant tree. Here are some of the fossil calibration points we used. We're always tweaking these based on basically what Ava tells me in terms of the most robust data we have for the most reliable fossils that we have for the rise of certain metabolisms or lineages that arose based on the rock record. So we looked at the last universal common ancestor, the evolution of oxygenic photosynthesis, the origin of methanogenesis, the origin of eukaryotes, and a few other things to anchor our tree in time. Okay, so we have a tree, a species tree of about 800 genomes. And then we take the gene tree of a specific enzyme that we are interested in. For example, the gene that catalyzes nitrogen fixation. So again, that's turning N2 gas into ammonium that brings nitrogen into the nitrogen cycle so life can use it. So we make our tree of life, and we make a tree of just that enzyme that we're interested in called nitrogenase. And we compare the shapes of those trees to each other. That's really all we do. And when we compare the shapes of those trees to each other, we can sort of figure out, okay, it looks like, these parts of the trees don't match. That means there most likely was a gene duplication at this period on the tree, or it looks like this gene was lost. And so it looks like the gene loss must have occurred at this point on the tree, or there was a horizontal gene transfer event, and we can figure out where that happened on the tree. So we call this reconciling phylogenetic reconciliation to try to compare the shapes of these trees and use that to try to figure out, okay, when were these genes gained? When were they lost? When were they born? And when were they horizontally transferred? So we focused on a set of genes in the nitrogen cycle. Some of the genes, we just didn't have enough data based on the data sets that we had to do this method. So I've highlighted those in gray, but we investigated all of the other ones that I have shown in black here to try to understand, when did these steps of the nitrogen cycle arise, and when did they start to proliferate across the tree of life over time? And what can that tell us about the evolution of the nitrogen cycle over time? So I'm showing you a bunch of histograms, and I'm going to go through this a bit more piecemeal, but I want to show you the whole picture to start, and then we're going to delve into some of the different pieces later on. Each of these histograms is oriented the same way. Again, time is on the x-axis, where the modern day is on the right-hand side, 4 billion years ago is on the left-hand side. So time is on the x-axis for all of them. The y-axis shows, it says here, proportion of events. So I want you to think about it as basically the number of times a gene was lost or gained or horizontally transferred or duplicated. So we're just looking at gene events on the y-axis, and you can see when gene events, and I should say there weren't that many gene losses. It was mostly horizontal gene transfer that you're seeing here. And actually horizontal gene transfer is highlighted here in green. So we can track when these gene events would have occurred based on the reconciliation I just told you about. I've highlighted what part of the nitrogen cycle these genes are part of using a color coding system. So along the top here, my cursor is gone, but along the top here, the ones highlighted in dark green are nitrogenases. So again, those are the genes that are pulling nitrogen gas out of the air, turning it into a biologically useful form. The genes in, I'll highlight the ones in blue, dark blue, those are genes related to denitrification from nitrate. And the genes along the right-hand side in sort of a lighter green are denitrification from nitrite. And there's some interesting patterns that we observed that we weren't really expecting that I'll highlight in a moment. But I'm going to start by focusing on the top 3 panels there. So those are the nitrogenase genes. And again, nitrogenase is the gene that catalyzes nitrogen fixation. So that's pulling nitrogen out of the air, turning it into a biologically useful form. Super important enzyme. And we wanted to know, when did it arise? When did it start to proliferate across the tree of life? One thing to know about nitrogenase is that it comes in 3 forms. It has 3 different metal cofactors that are part of that enzyme. One of them is molybdenum. That's probably it is the most common form of nitrogenase on Earth today. The molybdenum nitrogenase has a molybdenum as part of its cofactor, but there's also vanadium and an iron nitrogenase that has different metal cofactors within that enzyme. What we found was that the molybdenum nitrogenase seems to have arisen fairly early. We see the bars on this graph going back to about 3 billion years ago. which actually coincides with what Ava found in the rock record, which made her very happy. And in my experience, keeping her happy is very important. So I was glad that our phylogenetic data seemed to match up to what she was seeing in the rock record. That was a nice little gut check. And we also made sure not to include her rock record data in our calibration point. So it was an independent test of what she saw. And indeed, it does look like this form of nitrogen fixation arose fairly early. In contrast, other forms of nitrogenase, the vanadium and the iron forms of nitrogenase, don't seem to have arisen until later, at least according to our data. We don't have that many data points, so we're going to investigate this a little bit more carefully with expanded data sets, but at least on a first look, it looks like it was the nitrogenase, the molybdenum nitrogenase that arose first, and it was important very early on in Earth history. So what this tells us, again, based on our data, is that biological nitrogen fixation seems to have been an important source of fixed nitrogen in the Archaean, which makes some sense because you really need nitrogen. Like I said, it's an important limiting nutrient. You need somebody to supply that for you in order to support a biosphere. Some have argued that lightning, which does fix nitrogen, could have been sufficient to support an early biosphere, but our data suggesting that may not have been the case, that there just wasn't enough nitrogen fixed by lightning early on to support a biosphere. And so we saw nitrogenase arise fairly early. And again, our work suggests that it was the molybdenum version that arose first, even though it isn't thought that molybdenum was that widely available on the early Earth. Again, based on our data, which we want to investigate more closely, it does seem like even though there wasn't a ton of molybdenum, it was enough to support the rise of molybdenum nitrogenase on the early Earth. So that was kind of a cool thing we observed. All right, I'm going to bring you back to looking at the whole map of all of the data here, and I want to focus on the contrast between the graphs highlighted in dark blue, so along the bottom there, and the graphs highlighted in light green, so along the right side there. These are genes that are associated with denitrification, so taking nitrate as your source and then turning it all the way eventually into nitrogen gas. So here is the nitrogen cycle again. So we start on the upper right, so we have nitrate, and then it becomes nitrite, and then it becomes NO, and then nitrous oxide, and then N2 gas. That's the whole process of denitrification. It's very important on Earth today as an energy source for microbes. But what we found is that it seems like denitrification from nitrate, I'm going to go back here, those dark blue boxes seems to have arisen fairly early. It looks like they started to really proliferate across the tree of life around 2 billion years ago for denitrification from nitrate. So starting in the top left of this graph and going to nitrite. But genes using nitrite don't seem to have really arisen or proliferated across the tree of life until later, which was kind of surprising. And that includes the transformations from nitrate to NO, NO to nitrous oxide, and the nitrous oxide to N2 gas. So those three enzymes are shown in the histograms here. And you can see that we don't see a whole lot of action going on with these genes until about 1.5 or 1 billion years ago. We're still kind of trying to understand this. And I had to turn to Ava, my geochemist friend, to understand like what's going on here. I was literally hanging out with her in St. Andrews in Scotland last week. And she was like, I think maybe what was going on was that nitrite, oh, I wrote that wrong. Nitrite may have been consumed by iron. So Fe2, so ferrous iron may have been around in the Archaean and may have been consumed. And so it wasn't as widely available for the microbes. And so that might've been what was going on. And then after the great oxidation event, Fe2 became Fe3. And so we wouldn't have had as much consumption of the nitrite. So it would have been more widely available for microbes to use. That's one hypothesis, but we're not actually really sure. So this is an intriguing piece that we're still thinking about. So here's kind of the bigger picture, pulling back and looking at what our results showed, at least from this analysis, suggesting that nitrogen fixation seems to have arisen fairly early and was important in providing a source of fixed nitrogen to the biosphere fairly early on. It looks like DNRA, which is another nitrogen metabolism that is found in like marshes today, but is, could be more widespread than that, seems to have been fairly widespread more early on than we had thought. But then I wanted to also highlight the fact that denitrification from nitrite and came later than denitrification from nitrate. So different steps of the nitrogen cycle seem to have evolved later in a way that we weren't quite expecting. This is still very much, we did publish this, but we're still investigating some of the details of this, especially thinking about nitrogen fixation. So if there's ideas or thoughts, I'm happy to include that as we start to, refine our method and look more closely at some of these pathways. Oh, one more thing I wanted to mention, I'm going to go back here. I mentioned that nitrous oxide would have been an important biosignature, and that is one of those genes. The production and consumption of nitrous oxide, we don't think would have been it didn't proliferate across the tree of life until much later. And so that's important to know as we're thinking about, which biosignature gases would have been produced on an early Earth over time. It seems like that may not have been something that would have appeared until after a planet becomes more oxygenated, at least according to these data sets, but obviously more work is needed to think about this. So that was the nitrogen cycle. One thing we're pivoting to lately that we've been having fun thinking about is the evolution of the biological sulfur cycle. Sulfur is important for so many reasons. It's one of the most important electron donors and acceptors beyond oxygen. And so it's involved in many, many metabolisms. It's thought that sulfur reduction would have been important as one of the earliest metabolisms on Earth, sulfate reduction. And so we wanted to understand when these different steps of the sulfur cycle may have evolved over time. And we also paid careful attention to these cycles shown in red here, because those are genes involved in organic sulfur cycling. So creation of creation of molecules like DMS and DMSO, because our astronomer friends tell us that those would have been potentially detectable biosignatures. They're also cool because some of these are cloud condensation nuclei, and so could have affected the climate in that way as well. So we wanted to understand, when did these different metabolisms or the ability to create these molecules, how did that change over time? This is still, we actually just submitted this, so I'm only going to show you a little bit here. It's kind of I'm sure we're going to be changing it according to the vagaries of peer review, but what we broadly found was that it does seem indeed, as was previously thought, that metabolisms related to sulfate reduction, sulfur oxidation probably arose fairly early on Earth. But genes related to thiosulfate oxidation and reduction, so specifically the SOX genes, if any of you are familiar with those, we find them all the time in hydrothermal vents. Those don't seem to have really proliferated across the tree of life until a little bit later after the Great Oxidation event. So as the species of sulfur changed because of the redox state of the Earth, we saw different forms of sulfur metabolism start to arise. And organic sulfur cycling, the ones involved in DMS and DMSO production didn't arise, at least in the microbial world, until a little bit later, it looks like 1.5 to 1 billion years ago. So again, these biosignature gases, at least according to this analysis, and I should say we ignored the eukaryotes, which is a big source of DMS and DMSO, but the microbial side, it seems like they weren't producing the genes that are associated with the production of those molecules didn't really proliferate until a little bit later in Earth history. So that was sort of an interesting thing to observe. So basically to wrap this up, kind of on the early side, which is cool, we think, according to this analysis, that biological nitrogen fixation seems to have arisen somewhat early in Earth history, specifically the molybdenum version of nitrogenase. DNRA is perhaps more ecologically important early on than expected, and nitrite may not have been really exploited on the early Earth until later in Earth history. And that has potential implications for biosignatures like nitrous oxide, which is laughing gas. And that may not have been something that really was metabolized until a little bit later in Earth history, at least according to this analysis. And the bigger picture to really draw back here is that by combining our phylogenetic and geochemical approaches, so basically by teaming up with a geologist and thinking hard about, okay, what do we know from the rock record? What can we figure out from the genomic record, and how do these compare to each other? That's been a really fruitful collaboration. I've had a lot of fun working with Ava and other geochemists to think about how to interpret our data sets. Do they make any sense? And then giving this to astronomers who then look at us and say, okay, well, what does this mean, right? And then trying to interpret this so that they can then feed that into their biosignature models. It's been a lot of fun. So like I said, find yourself a rock friend or a biologist friend or an astronomer friend, because it can lead to really cool collaborations. I will end by putting up my acknowledgements. I will repeat that all of the work done here, for the most part, was done by my undergraduate students here at Carleton. One exception is I have a postdoc with Ava, Joanne Bowden at St. Andrews, and she's been sort of helping take over the building of some of these trees, which has been awesome. And funding for this came largely from NASA and the VPL and Nexus. And I'm happy to take questions.